Hello, everyone, and welcome to our Researcher Live webinar today, Empowering Cancer Researchers, Cutting-Edge Tools for Immunotherapy and Discovery. We're very excited to have Karina Chavez, a scientific researcher and an authority in the field of immunology, with us today. She will illuminate the latest advancements in this dynamic field and showcase tech stack to expedite the research and development process for novel pharmaceutical treatments in this essential sector. So welcome to our webinar, Karina. My name is Julia Heason. I'm the Senior Growth Marketing Manager here at Research Solutions, and I'm the moderator for today. So let me cover just a few quick housekeeping items before I officially hand it over, and we'll dive right in. So in case you want to rewatch a specific part later, share it with your colleagues, or you need to jump for another call, this webinar is being recorded, and I'll send out an email probably an hour after the webinar is over. So you have the recording, you don't have to worry about that. You can add all of your questions either in the chat or in the live Q&A section down below if you click on Zoom. We'll have dedicated time at the end of the webinar to get all of your questions answered. So feel free to join them either now or later in that specific part of the webinar. And then just another heads up that you're all in listen-in mode, so you don't have to worry about background noise, background whatsoever. So yeah, no worries about that. Now, for the ones of you that aren't familiar with Research Solutions, the company behind this Researcher Live webinar series, let me just cover a little bit. So Research Solutions was founded in 2006, and we provide solutions that simplify and streamline how organizations discover, acquire, and manage scientific articles and data. More than 70% of the world's top pharmaceutical companies, emerging life science firms, and other R&D-driven companies rely on us for one-click access to scientific literature. I'm also really excited to let you know that Research Solutions has recently acquired Resolute AI, an advanced search platform powered by AI and natural language processing. And then as of last week, we acquired Cite AI, an award-winning search and discovery platform that leverages AI to increase the discoverability and evaluation of research. So as you see, we're continuously adding relevant technology to our product family and to our product roadmap to make sure that you, our clients, are at the forefront of innovation. You'll actually get to see a little bit of that today as well. So now with that being said, let me hand it over to you, Karina.
Thank you, Julia. Well, welcome everyone. Today we're going to approach some of the tools that we use, or that my team is currently using to innovate and to get more knowledge about the cancer immunotherapy trends. So today we're going to discuss a little bit about some very interesting information and to learn about these tools that are boosting our innovation process. So the overview for today, we're going to cover a couple of topics. The first one will be cancer and immunotherapy basics. Then we're going to go to some recent scientific advances in this field. Then some of the landscape in the market and innovation, specifically for immunotherapy in the pharma world. Then we're going to have a little bit of sneak peek of the research tools that boost innovation that we're currently using. And finally, we're going to end with some round of questions and answers. So let's get started. Some people ask me, why do you doing research for cancer? Why is it so important? There's lots of diseases there. Cancer is a very group of interesting diseases, and it has a lot of impact in the economics of the world, and also obviously in the health of the human beings. So according to the World Health Organization, we have that cancer is the second cause of death around the world, just below cardiovascular diseases. These are statistics and numbers for this year. And as you can see, most of the people around the world that die due to a condition mostly are because of cancer. Cancer has been almost always on the top 5 causes of death around the world. So that's the reason that it's very important for us as researchers in the health science field to try to understand how this type of disease are behaving and how we can improve current therapies so they work better on the patients and they have the less possible of side effects. So what is cancer? Cancer is not like just one disease or one condition. It includes like a collection of diseases that have one thing in common. The one thing in common that these diseases have is that they have cells that grow uncontrollably, and these cells are spreading in other parts of the body. This specific like feature that cancer has to spread is mostly the reason why the patients that have cancer are dying. They spread to other tissues, they're spread to other organs and systems, and these systems stop working as they should be, and well, the patient deteriorates and they can die. So currently, the therapies that we have for patients that have cancer, they rely like mostly in the traditional ones. These include surgery, which is kind of like mostly the first option for the patient. It will depend, in the stage of cancer that they have, how advanced it is. And surgery is very likely accompanied by chemotherapy and radiotherapy. So these three are like the first options for patients. However, these, especially chemotherapy and radiotherapy, those two types of treatments are not specific to cancer cells or to the tumor. They will attack and they will kill all the cells in your body that are dividing, that are growing. This includes cells for your immune system. This includes cells from your epithelial tissues that are the ones, the type of tissue that is continually renewing itself. So these patients are going to have a lot of adverse effects. They can even deteriorate to the level that these types of therapies, chemotherapy and radiotherapy, can cause the death of the patient. And the patient won't die because of cancer, but because of the side effects of these types of treatments, because they are not specific. So taking that into account, researchers in cancer, we are always looking on options, to try to treat this type of diseases that are more specific. And these include, some of them include, like in big groups, hormone therapy, target therapy, which can include like gene therapy, and also immunotherapy. So in this specific Researcher Live episode, we are going to focus on immunotherapy and why some of the research in cancer is focused on immunology. Well, the natural way our body works is that we are constantly, maybe every day, generating damage in our cells, in our tissues, because of environment factors, because of infections with viruses, diet, etc. So our body naturally, the immune system has the ability to identify these cancer cells, as you can see. These cancer cells have very specific changes in the proteins that are conforming the cell. So these changes, our immune system is able to detect. They detect these changes. They can identify when a tissue is trying to convert into a cancer, into a tumor, and they will attack this tumor and they will kill it. So this is the way that regularly our body, our immune system is working. However, the tumor cells can evolve. It will depend on the type of cancer, the type of tissue that it's damaged, and the type of damage. They can evolve and they can like evade the immune system that it's daily protecting us. So when they, like they break the stage of equilibrium, they got to the escape stage. In the escape stage, they are able to totally evade the immune system and keep growing, or even spread to other parts of the body. However, with information that we have about how our immune system works, which are the cells that are the main participants in this type of protection in our body against cancer, we can take that knowledge and we can make some changes in the cells itself. We can even try to make some gene editing. We can also synthesize outside the body some molecules that are functional in the immune response against a tumor. We can build also antibodies and viruses, and we can put them inside the patient to try to fight cancer in a very specific way. So these are some of the groups that we can gather for cancer immunotherapy that are currently being in study, that we currently have some of these products clinical trials, and even so, some of these products are also being approved by FDA and are sold as like this new therapy for patients with cancer. So for these groups of different treatments or different options for immunotherapy in cancer, we have that monoclonal antibodies and immunomodulators are the ones that are most used currently in the commercial use for patients. Okay. This is very important, talking about maybe like competition, like how the trends are in the market right now. On a study that was performed early in this year by Drug Discovery and Trends, we have that at least for the pharma drugs that were more sold or best sold on the year, we have that at least 50% of all these drugs are monoclonal antibodies. And 90% of these drugs are for the use against cancer. So we have here the most important example, which is Keytruda from Merck company. This is a monoclonal antibody that is used against a couple of types of cancer, solid types of cancer that are very resistant to traditional therapy, including hormone therapy, including chemotherapy and radiotherapy. So as you can see, for Merck, they have sold around $12 billion, at least for the first half of 2023. And this trend is supposed to be improving. Around 2025, we are expecting a growth around 20%, 25% from these numbers, from these statistics. And well, here are just some examples of immunotherapy that has been used in pharma right now. We have Keytruda, we have Dupixent, we have Stelara, we have Gardasil, and these Humira, and these are all products that are synthesized in pharma labs by innovation. So as you can see, this is very important, very interesting in a way that if we want to understand how competition is behaving. So those are examples of what is currently like very studied, and we know that how it works, and that has been applied to patients commercially. However, if we go to literature and we do some search on what are the oncology breakthroughs of this year related to immunotherapy, we will find out that the therapy that is more currently being innovated and it's been in a lot of study and in a lot of focus from a lot of institutions and pharmaceutical companies, are the CAR T cells immunotherapy. And I'm going to tell you just a little bit about what this type of therapy is. So this type of therapy is not like super new of discovery. It was discovered, at least at the basics of how it works, in 1988 by some researchers that were aware that we have cells in our immune system that are called T lymphocytes. These cells are able to recognize these tumor cells, and while recognizing these tumor cells, they are able to kill them. They're very specific. So they, isolated these types of cells. Obviously, they started doing these types of experimentation on animal models and in vitro. So they noticed that if we can make some sort of genetic manipulation or genetic engineering on these cells, we can make them more specific and more functional against these tumor cells. They start running these edited or engineered T cells that are called CAR because of chimeric antigen receptor, which is the one that it's, this protein is the one that has been engineered or changed. And these CAR cells were start to be applied into experimentation. However, they have seen, these researchers saw that they were functional maybe in a controlled environment while being used in vitro and in animal models. But when they were like confront or put against like a real scenario of a tumor in advanced cancer disease, they were not working as they expected. So doing some more research, learning about the cancer environment, microenvironment, learning that the cells, the immune system cells also need some sort of communication between them so they can be activated and they can perform their killing functionality successfully. They were engineered and they were modified and improved in a way that right now in 2023, we have 4th and 5th generation of this type of therapy, which not only include the chimeric antigen receptor improvement, but they also include some engineering on cytokine release. So they will function better, specifically on solid tumors, where we have seen that this type of therapy is having some struggle working. Okay. So this is just a general visualization of how this type of therapy works right now. This is the advantage of using this type of therapy is that it will hold like specifics for the patient cells. They are going to be they were going to have a sample of blood take, and in the lab they will isolate the lymphocytes. These T cells were going to be genetically engineered. Once they are engineered specifically with the antigen for the patient tumor, they are going to be again infused into the patient. These cells are going to now in the patient be able to identify specifically the cancer cell and kill it, okay, destroy it. So this is like the main way that this type of therapy works. As an advantage, it will kill only the cells that express that specific protein, and it will not attack other parts of the body of the patient. However, this type of innovation had some side effects that were not something that we were willing. Some of the side effects, while the cells were destroying, the target, this destruction or this damage into the tissues was causing a lot of inflammation in the patients. So for this reason, from like around 5 years to now, there has been some development in this engineering of the cells, having evolution for best functionality. Specifically, as I just said, in solid tumors, we have these examples: TCR-like CARs, TAC T cell therapy, the drug therapy, AB TCR T cell therapy, and STAR TC therapy, which include modifications so they are better working on inflammatory type of environment in the patient. So for us as a research group, it's very important to keep up to date on what it's important to be researching, and also what it is important for us to focus so we can keep innovation, you know, in our institution, that we are not replicating some work that somebody else is doing, and that the work that we are doing, or the research that we are doing, it's focused, or it's well organized, so we can grant that our results will be successful. And for this, for us as a researcher, we can use some tools that are going to help us boost this type of innovation. And I was willing to share with you how you can use them, the type of advantage that they can offer you, and how easy is for you to get very, very, very interesting insights while using them. So I'm going to start sharing how they are used, but I first wanted to like organize these thoughts first. So the way that regularly we work at the lab is that we need to search and discover information that is related to the topic of interest that we are working on. So after that, once we gather all the patents, the clinical trials, the publications, etc., all this information, we will need to process it. We will need to read it. We will need to extract information that we need for, like insights and discussions, or for adverse effects, or very specific tweaks on methodology. So we then perform this innovation of the data analysis for innovation. Once we perform all these data analysis, and we extract and summarize all the information that is more important and more functional to us, we usually create reports. We can create publications. We can create content. And finally, with all this information, we can create our very own knowledge base, which we can also manage this amount of information for our benefit. So for all of these stages that I just shared with you, these tools can help us in each of these stages. So I'm going to start. The types of tools that I'm going to share with you today are Article Galaxy, Site, and Resolute AI. Okay, so sorry, let me switch to my let me switch to my browser, and let me give a sip of my coffee because I talk a lot.
Yeah, of course. Anyone, feel free to go ahead and grab yourself a cup of coffee, something to drink at any time.
Okay. Well, let me share you my screen. And can you see my screen?
Yep, that's good.
Excellent. So, well, as I was talking, first I'm going to focus on Article Galaxy in the first stage, which is going to be search and discovery. So for this stage, what we usually do is we usually look at the web for searching, like Google Scholar, or just like a Google, We also usually we use PubMed, so we can search for new information about specific topics. So I'm going to use the CAR cell therapy query just as an example. And as you can see, let me go back to PubMed so you can see how it works. So as you can see, there it is. As you can see, I have my both tools. I have the browser extension for Article Galaxy, and I also have my site extension. So in this case, both are very useful for me because from the side of site specifically, I can have a view, you know, of like the relevance of this information that I am willing to acquire, or I am willing to read, you know. They show this type of like images or icon, I call it batch, and it shows you the number of citations or the number of publications that have cited this specific research. But most interestingly, they also show you, or they share with you, the number of citation statements that talk about this specific research positively or contrastingly. For example, if I am using a very specific methodology that was described in this review or in this paper, and maybe doing, you know, like the experimentation in the lab, it's not working, and I don't know why it's not working, because it was published there, and I am using that references. So if I am looking on the number of the contrasting information that site provides me, I will be aware if someone else has published that their methodology is not working as they stated, you know. So this type of information can help me filtering which information might be relevant to me, what it will be more useful, you know, for me to read and to acquire. So this is like the view that site give us. If you click on the specific item, they can also show this this bar that you can hide, which will include the same type of information. Okay, and for the browser extension, it locked me out. Okay, for the browser extension, it shows you that specific page that information has been found, the type of access that you have, the abstract, and if you have it already in your library. So I'm going to go on the main search so I can show you. For example, if you are just like looking on Google or Google Scholar, this browser will auto detect the type of results that you have, and it will show you if these are articles that you are able, you know, to access. So in this case, here it found 10 articles, and I can see this list of information. Let's wait a bit. And as I told you, you can see like the most important information about that citation specifically, the type of access that you have, and you can save it for later in a list for saved. And you also have the site batch. So you can see publications that have cited this specific research, how many are supporting them, how many people are mentioning this specific work, and how many people have contrasting statements about this specific research. So this is for the Article Galaxy browser that allows you to access this information. If you are willing to, I'm going to go to site. If you're willing to get more specific information on site, you can click the batch, and it will display all the snippets that have a hit for your query in what part of the publication or in what part of that scientific content that specific query is mentioned, and the type of statement that it has been mentioned. So as you can see, I'm going to look for one that has contrasting type of mentioning statement. Oh, everybody is very positive. So let's see. I'm going to do a search here. On site, you can also do searches. So I'm going to close this one. And I'm going to search here. And as you can see on the snippets which part of the article or the scientific content it is mentioned, the confidence of the mentioning, and also if it's a positive statement or not. So I am just looking here for an example. Let me see if I can find something that it's negative. For example, this one. You can click on it. And you can, like read and review the type of information that the people is saying, the researchers are saying about that specific article. Okay, you have like the part of the article that they are mentioning. So this is very cool. For us, it has been very, very useful when we are looking for methodologies. And we want to make sure that methodology it's working, for other people. So we won't waste time and we won't waste money trying to replicate those specific results. So this is something that you can do with Cite. But my favorite part of Cite that might be also useful for you is the assistant. So I'm going to go to the assistant. Here in the assistant, as you can see, you can do a lot of stuff. My favorite part is that you can ask almost anything. And the AI will give you all the information that they find in the database, summarized in a way that is understandable, but not only that, they will also add the respective reference where they are like supporting what they are summarizing. So for instance, I'm going to write a question about what is our immunotherapy, and it takes a bit. It will depend also in the complexity of your question, but it will always, like generate this response. As you can see, it is composing a response, specific response for my question. And what I really like is that they give you the references where they are, like supporting what they are gathering in the summary. And not only that, you are also able to download the references. Okay. And what I also like is that there I can go to Article Galaxy. I'm sorry. I can go to Article Galaxy. Article Galaxy, besides, like helping you to get to know the type of access that you have, giving you reference information, and how relevant is this information for you to acquire. They also help us to manage our references. So here in my library folders, I already have my project for CAR T-cell therapy for the review that we were working on. So here I can add my citations. I am going to borrow this one, which is the one that I already download inside. And they are automatically uploaded in my folder. So when I am working on my report, as I shared with you, for example, I am writing, we were writing this article, and I am interested in having this information, these citations here. So I will only use the Article Galaxy Cite and Write tool, which connects directly to Word. And I can have access to all my folders in my library. As you can see, I can select a given citation. Okay. Here I can even see how relevant this information is. Okay, with the cite badge. And I am able to insert the citation in a long variety of customized styles or citations. Okay, so you just click in the text, you select the citation, and you insert the citation wherever you need. So I really like that because it makes all the process of searching, discovering, analyzing, and using that information for content very streamlined. So, okay, I'm going to jump now to another tool that I really like, which is Resolute. This type of tool is something that I really like when you work with a large amount of data, and when also you want to do some exercise of discovery and innovation. This type of platform, this type of product, what offers distinctively versus the other tools that I use is that they have a very large set of data sets or databases that they consult while you are doing your search. And they can connect all the results that you are looking for in a way that it makes sense to you. They also use, as the name says, artificial intelligence to make this analysis. And let me show you just a little bit of how it works. So I am also looking for trends and information about this therapy that is very breakthrough on cancer immunology. As you can see, we have publications, Crunchbase, patents, compounds. Some of these databases are very related to medical devices and drugs for FDA, tech transfer. Even we have key opinion leaders in case that you're interested, in getting to know what someone specifically is working on. So yeah, you have a lot of information. You just select the type of information that you are interested in looking for specifically. Maybe I'm interested in clinical trials. You can also do some filtering in case that you have very specifics. I want to have only results for T-cell or CD19, etc. And what you also can do, I'm going to select this one, is you can connect this information to discover more information. That's the thing that I really like. You can identify the people that is involved in a very specific type of research, where they are doing this research, if they are collaborating with another institution, the number of publications that they have related to that specific type of query, etc. So you can interact with this type of graphic, and you can download the information. The other thing that I really like about Resolute is, as I told you, you can do data analysis of large data sets. So in the case that I am interested in looking for these publications, I'm going to look up for again, and it's going to show me a number of results. I can filter these results as they are interesting to me. I can select them. Okay. For general purposes, I am just like showcasing how it works, but in a regular way, you will look for them, look through them. So you can select them. You can create your own collections. So I'm going to name it this Okay. And I can go to my collections. Okay. It's this one. Okay. I can see information specifically for my collection. I can see some trends. And this will depend also, you know, in the number information that you are saving. And you can also run reports. So in this case, I am going to run a report to analyze the technology landscape for that specific query. And it will generate you a report specifically for those items that I was interested in analyzing. So in the case that, for example, you are working along with your team and you have like patents, you have clinical trials, you have a lot of publications that you are willing to analyze, that you are willing to read, that you are willing to extract the most important information, but you maybe don't have a lot of time, enough time, you know, you have a short deadline to get that information summarized. This technology will help you to take all of that information and make it for you. So it is easier for you to, you know, like understand all the information that has been summarized and to work with that information. So it will depend on the number of records or the number of items that you are entering to analyze. It takes some time because, as you know, if they are patents or if they are clinical trials, even publications, you know, they are like a very large amount of data. So it is like reading everything and making this summary. But well, for just going to show you one that I made the other day. So I won't take a lot of time. I'm going to stop sharing and I will share it. Just let me know if you can see my screen.
Yeah, we can see it.
Okay, perfect. So once the process is done, generating, you know, the report, as you can see, what I really like is that it takes all the information that you select, and it creates this type of document. It gives you the executive summary, it gives you the methodology, it gives you the introduction of all the data that was analyzed, the industry overview, I'm sorry, for that specific query, technology categories, the technology profiles. It gives you also a market analysis, the impact on industry, the challenges and risks, and also a conclusion. And they also add all the references that were taken to generate this report. So as you can see, if you have a lot of information for a very specific topic that you are doing research on, and you want to generate content, you want to generate information for all the people that is working with you, you can run this type of analysis and just share it with your colleagues. So this is something that I really like about Resolute. They have some other types of data analysis for collections. As you see, they are let me show you my screen again for Resolute. They are not only, here it is. They are not only using this, you know, for publications, but, you know, if you are interested in doing some sort of like competitive analysis in the market with another company, with another institution, if you want to know how are the trends for the specific product that you're developing, if you want to know who is better to collaborate with, you can use this tool and it will make all of the analysis for you very, very fast. So these are some of the tools that I really like and that have helped us in our team to improve our innovation process. to make it more easier, to make it more faster. So I don't know if you have any questions. I was very excited to share this with more colleagues, so they are aware that this type of tool exists, and they are, you know, they have the benefit of using them too. So I think that's it from my side. Let me go again to share my PowerPoint.
Yeah, thank you. And we do have a couple of questions. If you haven't had a question yet, you can do that right now. Before we get to answer your questions, just wanted to let you know, if you're an existing customer, either using Article Galaxy, any of the other products that you started today, but you saw something that you don't have access to yet, you can either reach out to your account manager and ask them on how to get that added. If you don't know who that is, or you're not a customer yet, I'm adding a link in the chat right now. So you can head over there, and then it'll automatically connect you either to your account manager or to someone else on our team that then can help find the best person for you to speak to. We do also offer free trials. So if that's something you want to play around with, just reach out over the same link and ask for your free trial for either of the products. So under that same link, you can ask for Article Galaxy, you can ask for Site, you can ask for Foundation, the product of Resolute that Karina just shared with you. So yeah, just let us know. Okay, now let's dive into your questions, which is always my favorite part of the webinars. So the first one we received, I think, was related to one of the first slides, Karina, that you had shared. The question is, what country or countries are these considerations being considered for, and what are the biostatistics and biometrics of the incidence of cancer in locations affected?
I'm not sure if you mean like the stats for the number of deaths. It is according to the world. Okay, if that's so, yeah, it is according to what it's reported for World Health Organization. So it's what they include in their data. All the countries that they include are the ones that are reflected on those stats. Mm-hmm.
Great, yeah. And of course, if you have a follow-up question or you did refer to something else, feel free to either let me know in the chat right now, or we can also take this, of course, offline and then, yeah, share the exact details with you. Awesome. Thank you, Karina, for answering that. The next question is, can you discuss any ethical considerations or hurdles that arise with the use of AI in cancer research and treatment?
I'm sorry, can you repeat the question?
Yeah, are there any ethical considerations that arise with the use of AI in cancer research?
Well, as you may all know, guys, this AI approaches for science and for biotech, it's very new. It's something that it is still in discussion. What we can take advantage of right now, it's for data analysis, regarding analysis with references and with information that it's currently published. I mean, what I'm referring is that not trying to create new information. Maybe that's what you're referring to, but just help us with the AI to analyze big amount of information. I think that's the best approach that we can have with AI related to research, to help us to analyze this information and make it more faster, make it more easier to digest this big amount of data. Yeah, that's what I think. From the ethics point of view, I think it's something that it's still in discussion, how how convenient or how trustful will be to generate maybe new methodologies, or maybe create new information or new knowledge using AI experimentation or in silico experimentation. That is something that has been on discussion, but for this specific use, it's only for analyzing big data sets of information.
Yeah, huge question, huge topic, of course. I think you answered that brilliantly. Thanks, Karina. The next question, and what it looks like last question for today, is the references part that you showed. Is that something that you can share? with other colleagues, or is that individual use?
Which one? The one for site? Yeah, I mean, you can download the references that you are generating to the assistant, and you can use them as I show you, you know, for creating content, sharing with your colleagues. And from the side of Article Galaxy, I am going to share again my browser. From the side of Article Galaxy, there's a section where you can wait a second, so that it moves everything. Zoom. Yeah, there's this section on your references view. You can have your individual references for yourself and, you know, your research, your work, and you can also have share folders where you can share this type of content or specific content with your colleagues in your institution, on your specific team, etc. Yeah, we have here like lots of different features where you can work by yourself, or you can collab with no issue.
Awesome. Great. Awesome. Well, thank you so much, everyone, for asking your questions. If any questions come up afterwards, which happens sometimes, you can, of course, feel free to reach out via email. Those will get right into my inbox, or reach out over, you know, social media, email, whatever you prefer. And then we'll be happy to answer any questions that come up later and take them offline. Well, thank you, Karina, for, walking us through all this tech stack and giving us an overview. It was very insightful, and thanks everyone for joining. And I hope to see you around for the next webinar.
Thank you. Thank you for the invite.